A very good evening and welcome to one and all present here for the international webinar series DENTOPHARM 2022 on topic antimicrobial and anti-cancer potential of Piperlongumine extracted from Indian long pepper. I am Dr. Ramya, part of Dental Education Unit and Senior Lecturer at Sri Balaji Dental College and Hospital. Will be highlighting you about this program today. I would like to welcome our chairperson, Dr. Sri Nisha ma'am, Registrar Dr. Guminathan sir, Principal Dr. Jimson sir, and Dr. Sumati Jones ma'am, Head of the Department of Pharmacology at Sri Balaji Dental College and Hospital. Over to Sumati ma'am to share few words on today's webinar.
Good evening. Welcome to our DentoPharm webinar series. Today we have Dr. Lakshmi Narayan with us, who is well versed in antimicrobial and anti-cancer research. Today he'll be delivering lecture on antimicrobial and anti-cancer properties of long pepper. Over to Ramya again. About sir, please.
Just a little announcement on housekeeping before we get started. So if you have any queries during the presentation, please type them in the comment box given below. Now, I warmly welcome our guest speaker, Dr. Lakshmi Narayanan Rawat, who is a postdoctoral research fellow at the Division of Nephrology in Boston Children's Hospital and Harvard Medical School. He is an alumni of Jivaji University, Gwalior, from where he obtained his BSc Biotechnology and Masters in Molecular and Human Genetics. He completed his PhD in Biological Sciences under supervision of Professor Dr. Vijayashree Nayak on anti-cancer potential of piperlongumine and ursolic acid in human cancer cell line in vitro at BITS Bilani, Goa. He has research interest on human genetics and molecular diagnostic clinical research, cancer biomarkers, and therapeutics. He has also worked in various projects as Scientist D in a project of COVAXO and COVID-19 vaccine trial at ICMR National AIDS Research Institute in Pune. And he was also Junior Research Fellow in DST sponsored project over the years. Later, he moved to the United States to pursue his certificate courses on clinical pharmacology and clinical research from National Institutes of Health, USA. At present, he is working as postdoc in Boston Children's Hospital. He has participated in numerous conferences and published his studies in journals with highest impact factor 6.5. Also received Young Scientist Best Poster Presentation Award in IVC MBBS 2020. We welcome you, sir.
Thank you very much, Dr. Ramya, and I'm thanking you everyone who is for inviting me for such a good lecture series. And I would like to thank the college principal and all the faculty members of the college and departments. Thank you very much. So I'll start then. A very good evening to everyone. I'm here to deliver a brief lecture on the antimicrobial and anti-cancer potential of piperlongumine extracted from lung pepper. During my PhD, I have worked on two herbal compounds, which one of them is piperlongumine and another one is ursolic acid. So piperlongumine is very known to in modern medicine as well. And from the ancient time, we are using lots of traditional medicine practices. So I'll briefly introduce you about the traditional medicines. So what are the traditional medicines? So any kind of method which we use to treat the disease condition or the certain symptoms of the diseases. So those are called as the traditional medicines. So basically we use the phytochemicals or animal resources to treat the disease condition and the traditional medicine practices. So as we Indian, we have a long history of the traditional medicine practices. Like for in the whole world, we export almost 70% of the spices. Those are the fine herbs that have a lot of beneficial impact on the human health. And even in the modern medicine, we have several drugs which are derived from the natural resources like the plants or animals, like and the bacteria, fungus, and everything. So basically, these are the natural medicines without any further chemical modifications. So, for example, the Taxol and doxorubicin. So, Taxol is extracted from bark of yew tree, and doxorubicin is also an antibiotic from the fungus. So these are the primarily used chemotherapeutic drugs, like for any kind of cancer in the modern medicine. So that's how like we are doing that. We are using phytomedicine as even in the modern medicine practices. So because of that only, we have to understand that these that spices and these phytomedicine we are surrounded with are very beneficial. Just we need to explore them much more than the previous, because at that time we don't have that much of resources to explore the beneficial aspects of them, but now we have all the facilities to do that, so we should do, and we should focus on the modern medicine by keeping them together. So even in our home, we use lots of home remedies. Like in south part of India, there is a one home remedy, tirikadukam, that is a combination of these three components, like dry ginger, black pepper, and the long pepper. This is we use for to treat the respiratory problems, cough, cold, fever, and also this is very beneficial in the digestive tract problems. So that's how we are using, like in our daily life as well. We use lots of spices, and that is also maybe the case, like we are having a much better immunity comparative to the Western world. So we are today focusing only on the long pepper. So this is the long pepper in on the plant, but we use it as a spice, and mostly spices are we use in the dry form. So in even in the oral a problem like oral health, gingivitis or inflamed gums are the major problem. You must have known it like very frequently, and it is the most common, like in every person has suffer because of this kind of problem. So by over the, what is the reason behind it? So the overgrowth of bacteria actually is the that disturb the symbiosis, because we have a lot of microbiota in our body and as well as on the skin. So if we they live in the symbiosis form, like we are getting some benefits from them and they also getting food from us. So but if this balance disturbs, that will cause a problem, and because of that disturbance the immune cells infiltrate that particular tissues, and then they will cause the inflammation, and ultimately the inflammation will cause the damage to that surrounding tissue, and that again will form like create the problem. So sometimes inflammation is act as a antibacterial kind of method, but the severe inflammation can also favor the bacterial growth and propagation. So here, like gingivitis is the example of overgrowth of bacteria and the inflamed gums. So because of inflammation, they damage their own tissue. So how we can manage it? Like even in like we don't have the like people who don't have access to the modern medicine and modern therapy, so they can also manage these kind of problems with their home remedies. Like they can also prepare the uh, extract of Piperlongumine, which is known for the anti-inflammatory and the immune modulatory function. And Piperlongumine also has the antibacterial and antifungal properties, and there are lots of research article on that. Like piperlongumine is very useful for these kind of actions. And so in these this particular condition, we can use the anti-inflammatory and immune modulatory properties of piperlongumine to control the inflammation as well as the bacterial growth. So this can ease the symptoms and then can prevent the gingivitis to be more severe and to form the gum disease. So coming to the antimicrobial properties, like how we actually decide like any compound which is having the antimicrobial property or not. So in this case, we use the extract of piperlongumine and against a certain class of bacteria, rather like it is gram positive or gram negative. So these are the gram-positive examples of gram-positive bacteria like Bacillus and Staphylococcus, and gram-negative bacteria are the E. coli, well-known, and Salmonella typhi. So these are and some also pathogenic bacteria also we test in the the main purpose of this, that this compound can be used against those kind of bacteria to form and to prevent the disease condition. So these are the few methods to test how we prepare the extract. I'll discuss in the later slides. And these are few methods like time kill test and flow cytometric methods we can use for to test the compound. And this is the disc diffusion method. In this, we prepare the plate and we make few holes in that, and then we uh, put the bacteria to grow overnight. And then after the growth, we put the our compound of interest in those boards, and then it will form the zone of clearance. And then because of their antibacterial or antifungal properties, so we measure this diameter of the zone of clearance, and then we calculate the how much of concentration of that particular compound is being beneficial for the can inhibit the growth of bacteria. And also broth dilution, microdilution method, in that we serially dilute the compound as well as the uh, bacterial culture, so that we can precisely get the concentration as well as the number of bacterial colonies. And ATP assay is also one of the best assay to describe the antimicrobial activity against the bacteria and the fungus. So there are several articles, as I told you, like on the piperlongumine, they have done a lot of work and to establish the antimicrobial and antibacterial properties in the different kind of. But also like in modern class of medicines, we have in Ayurveda, we have several kind of products. Those are already approved by and the like for the gum problems, we have the toothpaste, for the cough syrup kind of, and chyawanprash to immunity boost. Like there are several others as well to use with the use of piperlongumine. So coming to the next part, like till now we are talking about the whole extract, like that is the crude form of the piperlongum, that is from the fruit. But now we'll talk about the more specific or more active form of the piperlongum, which is an active ingredient of the piperlongum fruit. So these are the Piperaceae family members, even the black pepper is also in that. So all of them has the piperlongumine or piperin. Both are the same class of molecules, and those are the active alkaloids. So that those have the amide moiety separate and the aromatic moiety separated by the aliphatic chain. So extraction part, in the next slide, I'll tell you how we extract. There are several extraction methods, like from raw to very specific one. So very crude one is like if you put the dry long pepper powder in the water, and if you boil it for a very long time, it will give you the extract of it. But we need the active ingredients of that. So first we need to may prepare the extract from the powder form of the fruit. Okay, so there are several methods, like oxidation is the very oldest method, and ionic liquid base extraction, like chromatography, ultrasonic extraction, microwave assisted extraction, and also supercritical fluid extractions. In that we change the fluids, like because these kind of drugs are more soluble in the organic solvents rather than just in the water. So we use either methanol, ethanol, and the DMSO to extract those kind of molecules. But if we are doing extraction, there is no only one kind of molecule. There are several others, right? So to purify further, we use the HPLC system or UPLC system, uh, high performance liquid chromatography, to precisely extract a particular compound. In that, so we run the standard, so there is a standard of it, and then we run our extracted compound. So in that we can collect this peak in the another vial, and then further we will uh, uh, make the dry powder of it by the. So coming to the active alkaloid of long pepper, which is called as Piperlongumine. So the most of the work I have done on the Piperlongumine, that is the pure form of the long pepper, and it is very known to treat the several diseases, like even for the viral diseases also it is very beneficial, other than the gastrointestinal and common respiratory problems. So my work is focused on the cancer. So I'll tell you more about that. So also what I have done in my PhD, and this is only the part of the work which I'm showing, because that is a lot to show. So the PL is reported to mainly inducing ROS, a reactive oxygen species. I'll tell you how ROS can be beneficial as well as the favorable create the favorable situation for the cancer. So this is a fruit, and this is chemical structure of Piperlongumine. So piperlongumine is already known for the anti-cancer properties, and the other biological effects also well studied in so many systems, like in vitro, in vivo, or even the higher organisms. So coming to the cancer part. So cancer is defined as an abnormal growth of cells which tends to proliferate in an uncontrolled way. So as we all know, cancer is a major problem nowadays, and the cases are increasing rapidly, and there is no ultimate cure for the cancer, because if certain patient go for the third and fourth grade of cancer, it is very difficult to treat it. Only you can manage it, but it is literally very difficult. So what is the reason behind the cancer? So the main reason is the genetics. So genetic mutation, alteration, epigenetic alteration, chromosomal aberration, and also other than that, there are several environmental risk factors, like the food. Our food is the very main factor in that, like consumption of alcohol and red meat, and also the smoking of tobacco and certain hazardous exposure of hazardous chemical compounds, the facilitator of the cancer. But the main reason is the genetic mutation, epigenetic alteration, and the chromosomal aberration. So I'll speak about the genetic mutation. So mutations are the any change in physical change in the DNA sequences, and we have the 98% of our genome is the non transcribable, like that you can call it, that is non-codable. So that doesn't code for any protein. So that we also called as the junk DNA. But that is not the junk DNA. There is some evolutionary significance of that DNA. So precisely the mutation in the there are two class of genes, like one are the proto-oncogenes and another one are the tumor suppressor genes. So the function of proto-oncogenes is to derive the cell proliferation, means they need for the cell division. But other part of the other class of the genes, those are tumor suppressor. Those are the breaks of like cell cycle. They don't want to cell to grow or to divide. So any mutation in those kind class of genes can result into the cancer. But not only the one gene can, because we have the two copies of each gene. So any there are certain genes which can cause the problem only after one mutation, and there are certain genes which can cause after the mutation in both the copies. Other than that, the epigenetic alteration, which is the nowadays it is very fascinating topic, like how we the cellular mechanisms, like how they can be change in the genetic expression of those genes rather than the just mutation or the change in the physical form. So, what happens in the epigenetic alteration? There methylation group, methylation of any DNA content can restrict the access to the polymerases that are responsible for the transcription. So ultimately, epigenetic alteration inhibits the transcription part and ultimately the translation. So there will be no protein. So ultimately, it is inhibiting the like gene function by just change in the form, like not the permanently but temporarily change in the genes. And the last one is the chromosomal aberration. These are the major changes in our genome, and they will lead to the instant effect, like you can instantly get the cancers or other disorders. So, because of these factors, normal cells transform themselves into the malignant cancer cells. But how we can distinguish them, like how we can there should be certain characteristic features on basis of that we can differentiate the normal as well as the malignant cells. So obviously there are certain hallmarks of the cancer which scientists has decided, like, okay, the normal cell has these one and the cancer cells have those one. So there are few main features of the cancer cells, like resisting cell death. So in normal cells, we have a programmed cell death, a phenomenon that is the within the cells we have a biological clock. So eventually, after subsequent cell cycles, so the length of telomeres will reduce after each cell cycle. So after certain cycles, like telomere will be that short, like they will recognize by the DNA damage proteins, and they will force the cells to go for the cell death. But in terms of cancer cells, they don't have such phenomenon. They have the telomerase activity. So they can synthesize the telomeres. So they can, because of that only, they are uncatchable. Like those proteins can't catch them to and force them to go for apoptosis. So because of that only, they are replicative immortal. So that's why we call them as a immortal. And also the evading growth suppressors, like as I told you, like the tumor suppressor proteins and tumor suppressor genes are which like they restrict the growth of the cells, but they also invade, they uh, purposely mutate them and restrict their activity. And also the metastasis is also is a problem in the cancer and hallmark of the cancer, because most of the cells we have are the epithelial in nature, and those are non-motile cells, but cancer cells are the mesenchymal kind of property, and they can move toward any site in the body, and then they can develop the tumor. As a tumor, there are rapid growth of the cells, and they form the bunch of or mass of the cells. And obviously, if something is growing very fast, they need much nutrients and the oxygen to survive. So, because of the hypoxic condition in the within the tumor, they'll produce some factors like vascular endothelial growth factor, that is VEGF, and a hypoxia inducing factor like HIF2 1 and 2. So, in response to that, our whole body start the angiogenesis. Angiogenesis is the formation of new blood vessels within the tumor. So to fulfill that nutritional requirement, they will form the new blood vessels within the tumor and provide the favorable condition for the metastasis. So these are the certain hallmarks of the cancer. And so what are the current treatment options we have for the cancers, and what are the drawbacks? Even in the modern medicine and the surgery, we have lots of developments to treat the cancer, but still we are developing a lot of therapies and modality for the cancer treatment. But there are certain limitation of every therapy and treatment option. Like not all the patients who need treatments are eligible. Like because their health condition doesn't permit them to go for such kind of harsh treatment, because chemotherapy and radiation therapy is a really really bad one. And uh, and they have a lot of side effects as well, like the instant side effects are the pain, infection, heart failure, nausea, vomiting, because these kind of therapies are very forced, like they are very random, like they don't have very precision in their application, so they can cause the death of normal cells as well. And long-term consequences can be heart failure and development of secondary cancers. And because of these secondary cancers, cancer patient has to go for the treatment again and again. So that is a very bad situation in the cancer. So we'll come to the hurdles, like what we are facing nowadays. Like even we have very good drugs, those can kill some cancer cells, but what are the problem? So the drug resistance is a major problem in cancer. Like we cannot give the like so suppose if the drug is have to be administered like 500 mg per 2 days, but we cannot give more than that, and it should not affect the other organs as well, like it should not be lethal for that person, otherwise the therapy is non-effective. So the problem with the lower concentrations of drug is that drug resistant, they will immediately develop the drug resistant, and this problem persists with the most types of the cancer. So there are certain reason behind it, like epigenetics. As I told you, they will like in the epigenetics, they'll change the axis of the DNA. So if that particular gene is non-accessible, we cannot do for they cannot translate that particular protein, and that gene is will be the non-functional. And drug inactivation is also another problem, because most of the modern medicine are in the form of prodrug. Those are not in the active form of the drug. So they need certain physiological condition like particular pH, particular enzymes, particular condition to activate that drug. And most of the drug activated by the liver enzymes. And also liver is also responsible to neutralize them and to prepare them for the excretion. And drug target alteration is also a very big problem, because we test the drug in the in vitro, in vivo, and in the higher organisms. But in the actual condition on the humans are much more complex than the other animal form. So in that as well, there are they have seen lots of drugs which are supposed to hit that particular target, but they are not doing it. They are also targeting some other uh, molecules, so that will also cause the problem. And epithelial to mesenchymal transition is also the problem in that can form the drug resistance. Cell death inhibition, DNA damage repair is also can lead to the drug resistance, which is a primary, nowadays it is a primary cause of the therapy failure. So other than that, like other than drugs, we have certain options, treatment options like cytokine therapies, like for the particular to reduce the inflammation or to promote the inflammation. We give the certain class of cytokines to uh, invade the immune system to the tumor. Like we provoke the immune system to kill the cancer cells. As cancer cells are not very different from the normal cells, because they also have the certain class of molecules which can be used as to describe self and non-self or foreign. But they also have those kind of molecules, and then they take the advantage of that system to for the immune escape. And so for that, we have monoclonal antibodies, like so many, and monoclonal antibodies nowadays are being used in the cancer treatment. But these are very expensive. Like not everyone can afford them, and though they are very specific, but there are certain again there are certain limitations of that. Like for the particular type of cancer, you need to have the certain class of antibodies, monoclonal antibodies, and you can't just give the monoclonal antibodies alone. You have to give in the combination. So that is also called as a combination therapy. We will also discuss the combination therapies in later slides. So our main focus is on the why we are using the herbal approach, although it is considered as a old kind of practices. But even as I told you earlier in the earlier slides, there are certain class of molecules and drugs which are extracted or developed from the natural resources, and those are very much effective against all kind of diseases, like specifically for the cancer. So, so actually, herbal approach has certain advantages over the synthetic drugs or the synthetic therapies. So they have broad spectrum effects. Like, and if you choose any class of molecule extracted from plant or animal source, those are don't target just one molecules, and they target a lot of targets to ease the symptoms or to manage the disease. And also those are very much effective and nil or low organ toxicity. The major problem here, like the dose implementation, like how we decide the dose of that particular therapeutics. So in that case, like herbal compounds are very, very beneficial, because they don't have as such toxicity to the organ, like vital organs like liver, kidney, and heart. Whereas the modern the synthetic drugs, like those therapies are very those are cardiotoxic, especially. So most of the patients who are going for the chemotherapy can develop the symptoms of heart attack. And even the and there is a one disadvantage of the herbal drugs, the dose, because herbal drugs are not that precisely effective against the treatment. So we can use them as a complementary medicine. There are certain drugs which we can use as a primary therapeutics, but the complement as a complementary medicine as well, they are very beneficial. So there are so many clinical studies reported, like herbal medicines are beneficial in terms of the combination with the conventional therapies. So they also not only increase the survival rate of the cancer patient, but also the quality of life. Like there are very few chances like you'll get the secondary form of the cancer. And also they can reduce the dose of the primary therapeutics like synthetic drugs. They can also, because they can complement the function, like even at the low doses, the efficacy of that particular therapeutics is equivalent to the higher doses if we give them in the combination. So my work is majorly focused on the intestinal and the osteosarcoma, but I'm discussing more about the intestinal because the idea about that is like whenever we take something as a food, like we are taking lots of spices and that goes directly to the intestine. Instead of that, the colorectal cancer is the third most common cancer all around the world. Not in India, but in India we have lung cancer is most dangerous one, because in India there is a big problem of the smoking. So small intestinal cancer, or you can call small bowel cancer, is very rare and account for less than 1%, whereas the large intestinal cancer or the colorectal carcinoma is the third most one, with high incidences and mortality. As we previously discussed, like family history, age, lifestyle, food, smoking, and alcohol consumption can facilitate the development of the cancer. So, and there are the treatment modalities are all the same for every cancers. Like most of the tumor can be removed by surgically and then followed by radiation and the chemotherapy. So there are certain kind of diseases which actually can favor the development of cancer, intestinal cancer. So coming to the main work, what I have done. So I have focused on the in vitro part of that work, and I used two cell lines in that, INT407 and HCT116. So INT407 is a small intestine origin, and HCT116 is from the colorectal origin. So after that, I gave the piperlongumine treatment in different concentration and did the following assays. So coming to the result part, so MTTS is the most basic method to determine the dose of that particular compound or drug. So here you can see there's a percentage cell viability, and these are the concentration of piperlongumine in micromolar at two time points, 24 as well as 48 hours. So you can see there is a pattern, like if you increase the time as well as the concentration, there is a decrease in the cell viability. That means like the availability of that drug is a major factor, and also the concentration. So we need to choose that concentration which is not very toxic and also not very ineffective. So by linear regression method, we use to calculate IC50 value, which is the inhibitory constant half inhibitory concentration, which is 13 micromolar for INT407 and 8 micromolar for FCT116. So as INT407 is from the small intestine origin, and this is more less aggressive kind of cancer, but SCT116 is more aggressive cancer. So that also like you can see that, like you can make the statement like so piperlongumine is effective against the more aggressor cancer, not the mild one. So that is good for us. So we choose these concentration for the further experiments, and all the treatments are statistically significant. That is a valid point in our kind of research. So coming to the next part, so the next thing we see, like so in previous method we just use the cell viability, how it is impacting. So but in this method we use the like how much cell death actually piperlongumine is causing. So here we use the two kind of stains, acridine orange and propitium iodide, which are very common. This is very common method to do that, and followed by the fluorescence microscopy. So here you can see the acridine orange has the property like it can go in any viable or the dead cells. But piperlongumine, that propitium iodide is very limited to the dead cells or the membrane compromised cells. So the main features of apoptotic cells is the membrane. It has like cells will have the compromised membrane, like they don't have the intact membrane. So propitium iodide can directly go and give the red fluorescence, and whereas acridine orange can go in all the cells and give the green color fluorescence. So if you see the in the control cells, all the cells are green. So that means most of the cells are viable. But when we are increasing the concentration of piperlongumine in both the cell lines, you'll get the more number of dead cells or more number of apoptotic cells. So that means, yes, our doses are working effectively. So coming to the next part, so as I previously discussed that piperlongumine is known for to induce the ROS. Okay, so because ROS has a dual impact on the cancer, like it can favor the cancer and it can also be used as a therapeutic modalities. So I'll tell you why. So these kind of conditions, like these kind of situation, like reactive oxygen species molecules, works in the concentration dependent manner. As in the low concentration, it acts as a signaling molecule that can promote the cancer invasion. So even if we have, because that ROS is an integral part of our metabolism, because our mitochondria produces the ROS frequently during the metabolism. So as the cancer cells are highly metabolically active because of their rapid growth and cell division, so they have higher level of ROS as compared to the normal one. So we are using their situation to kill, like it's kind of we are using their weapons against them, right? So the piperlongumine is known as a ROS inducer. So we are giving, and but in the higher concentration of ROS, so what will happen? So after certain threshold, ROS will act as a destroyer, like that can promote the DNA damage, and that can also, and in response to DNA damage, the apoptosis will happen. So we are quantifying the ROS by DCFTA staining. This is a fluorescence molecule reduced by molecular oxygen, that is called a reactive oxygen species. So in the control and the lower concentration in both the cell line, you can rarely see any fluorescence. So that means they have very less ROS. But in the higher concentration, as we increase the concentration, there will be increase in the ROS. Like this is a quantitative histogram representing the quantitative data of these images. So as we increasing the concentration, the ROS is also increasing. So it's again a plus point for us, like yes, this piperlongumine is working. Coming to the next part, as I told you, like the ROS, excessive ROS can cause the nuclear damage or nuclear fragmentation, and any cells with the nuclear damage cannot survive because they have to transcribe and translate proteins for them, and those are like transcribed from the nucleus. So because of that, like because of excessive ROS, they have fragmented nuclei in the higher concentrations. Like if you see in the control, the nuclei are very round and uniform. But in the highest concentrations, you can see the fragments of nucleus. Every nucleus is fragmented in the both the cells, and this also. And we quantified these fragmented nuclei, and you can see there is a significant increase in the fragmentation. So yes, definitely this is our goal to achieve that. So yes, we are getting that part, like we are can the piperlongumine can cause the significant amount of DNA damage. So coming to the next part, so the next property of cancer cells to invade the metastasis. The metastasis is the very crucial thing in the cancer development. Like if there is a only bunch of mass, we can remove that by surgery and followed by radiation. That is treatable. So because of that, the tumor is only the stage 1 and 2 of the cancer, but in a stage 3rd and 4th, after metastasis, they'll call that kind of cancer is the 3rd and 4th grade of cancer, and those are very lethal. So this wound healing assay to assess the how much like cancer cells has a property to migrate in even in the in vitro models. So this wound healing assay, we are in the monolayer, we are creating a single line wound kind of a structure, and we remove these cells and we wash them with PPS, and then we'll put them again in the media with our therapeutics. Like here we are using the piperlongumine. So we are using 3 concentration, 1, 13 and 20 micromolar, and this is the IC50 value of both of the cells. So if you see, as we are incubating, this is at the 0th hour, there is like when we make the wound. And after 12 hours, you can see there is a migration in the cells, and they can cover that area within 24 hours. So how fast they are growing in both of the cells. You can see in the controls, the wound is completely filled by the cells at the 24 hour. But if we treat them with the piperlongumine, and you can see in the lower concentration, it is effective, but not that much. They are still moving, but in the IC50 and the higher concentration, they are not able to move. They are very restricted, and even in the higher concentration, there will be no cells. So if we measure the size of the wound, you can see in the control the size is decreased, but in the higher concentration, the size of the wound increases. That means our therapy is working perfectly against the metastasis, against the migration property of the cancer. So coming to the molecular part of it, so these all physiological properties, they are controlled by the molecular molecules. So those are like we have to be very sure like this is happening on the cellular as well as the molecular level, like to be foolproof. So here there are certain class of genes. I'll tell you these BAX BCL2 survivin is the these BAX BCL2 is basically apoptotics related genes, and survivin is the growth promoter gene, and P21 P53 these are the cell cycle regulators, and SMAD4 is also a tumor suppressor protein, and FN1 that this is fibronectin 1, CDH2, CTNNB1, twist1. These all are the markers of migration markers. So we have to check them because we are performing those kind of experiments. So if you see in the histogram as well, you can go along with the readings. So in both the cells, BAX is the pro-apoptotic gene. If this will be high, there will be more apoptosis. And BCL2 is the anti-apoptotic protein. So that means it is getting down after piperlongumine treatment. So that is also, we can confirm that, okay, that apoptosis is happening. The survivin is responsible for the cell survival, that is also going down, and P21 and P53 SMAD4 these are the tumor suppressor which are going up. That means yes, we need that, there should be no cell growth in the cancer. And these also migration marker genes are also getting down, this is the fold change, like how much fold is going down. So if you see in all the cells there are more than twofold, like that is very significant. And even for some genes you can see there is a 11 or 13 fold of uh, reduction in the expression. So coming to the next part, which is a combination therapy I was telling about. So the combination therapies is which, like we can use our these herbal compounds in combination with the already approved chemotherapeutic drugs. Like I told you about the Taxol, that this is a modified form of the Taxol, that is paclitaxel, and which is a primary chemotherapeutic drug in all the cancer. So here we are using the piperlongumine and the paclitaxel in the combination, and this is also an in vitro study. So I treated the cells with 1, 2.5 and the 5 micromolar of piperlongumine, and 0.1, 0.5 and 1 micromolar of paclitaxel. So you can see this is the matrix of inhibition. So you can see there is no inhibition in like control, and highest inhibition in the higher concentration. And there is a pattern, like if you increase the doses, there will be more inhibition. And this is how we calculate the synergy score. That means both the drug are working in the synergistic way. They can be synergistic, they can be antagonistic, or they can be additive. So the additive is like they are working independently. The antagonistic means that those are working against each other, and the synergistic means they are working together to achieve that goal. So you can see this is a more synergistic doses this one, this area you can target. Like these doses are very effective against the studied experiment. So because also we can see the score, any A score in the positive is considered to be a synergistic. Any score in the negative, it is an additive effect. So this is a uh, dose dependent curve for the piperlongumine and the combination, piperlongumine and the paclitaxel and the combination, these you can see. And in this also, like here, if these are the dose effects, and these all the doses falls under the synergy category in this column. So that means all the doses like those combinations are the synergy and working in the synergistic way. That means these drugs can be used together for the treatment of cancer. So this data is for the INT407, and the similar data is for the SCT116 cells. You can see there is a synergistic a plot for HCT116 with the same concentrations and the 0.1, 0.51 of paclitaxel and 1, 2.5 and 5 of piperlongumine. And in this also, because this is different model and this is also different model. This is SHA and this is a different kind of software. In that we analyze the synergy or antagonistic or additive effect. So in this also, all the doses are falling under the synergy category. That means we can use these both drugs like piperlongumine and paclitaxel together. So coming to the conclusion of this work, as I told you, like piperlongumine can induce the ROS by targeting the mitochondria, because most of the ROS is produced by the mitochondria during the metabolism. And also, it also inhibit the antioxidant enzymes like TRX1 and other well-known antioxidant systems. Like because we our cells within, they have the both the things, because if there is a ROS, there will be antioxidant system or antioxidant enzymes. They can regulate the uh, ROS concentration within the cells. But what happens if there is a block in the antioxidant enzyme, and the piperlongumine promote the formation of ROS? So that the excessive ROS will cause the DNA fragmentation, and in response to that, there is a first responder of DNA fragmentation is the p53, which is also called as the guardian of genome. So because this is a very powerful tumor suppressor gene, and it will further upregulate the BAX, which is the pro-apoptotic gene. So that will recruit the other molecules to induce the apoptosis. And in another way, alternatively, the piperlongumine is upregulating the SMAD4, which can regulate. This is also a transcription factor, and it can regulate the expression of P21, which is a cell cycle regulator. And upregulation of this cell cycle regulator will lead to downregulation of BCL2 and survivin, and that will also lead to the apoptosis. So this is how this mechanism is working, and this signal transduction is working to induce the apoptosis in cancer cells. So coming to the conclusion of this work, so we developed we found out the IC50 concentration, which are very comparable to the primary chemotherapeutic drugs. And as piperlongumine is it's regulate these all machinery by inducing the ROS level in both the cell lines, and they can also cause the lethal oxidative stress to induce the apoptosis. Gene expression analysis further confirms that whatever we have done at the cellular level, and are able to revert. Also, it is very interestingly that this can revert the drug resistance by making cells more vulnerable for the apoptosis. And finally, that we have successfully tested the synergistic impact of piperlongumine with the paclitaxel. So that can this combination can further go for the in vivo and to treat the cancer. So other than that, there are several studies which are already being reported, like because as I initially told you that these class of molecules, they don't target just one molecules, rather they target the entire class of molecules. So these all are the molecules, or these are the targets which is targeted by the piperlongumine, and they ultimately induces the apoptosis. So that's what we need. Like instead of targeting just one molecule, it is always better to target several molecules. So there will be no chance of the cancer cell escape. And in this one also, like this is a different pathway. So also piperlongumine is one study has reported the piperlongumine can also uh, be very beneficial in the radiotherapy. Previously, we are talking about the chemotherapeutic aspect of it, but in the radiotherapy as well, it is working perfectly. It can also like limit the radiotherapy's side effects. So the future perspectives can be we are working on the extracts. So even the more precise formulation can be prepared and remedies can be prepared for the particular disease condition. As you are working in the like dental field, so you can also like I have shown you the all the toothpaste, those contains the piperlongumine. So you can also in future can work on the oral rinse or washes to made from the pipli extract. So those can also be very beneficial. Recently, I'm going through the literature, and I found out China has patented so many products. Those are made from the pipli extract, like piperlongum. So, but we are not doing it till now. So I don't know why we are not working on that. Uh, if you see the all kind of these phytomedicine, China is number one in the research. Those are working very in very good way uh, to to explore the phytomedicines. And also the nano formulation we can develop for further enhancement of therapeutic potential of these therapeutics. Like I have also worked on the gold nanoparticle mediated drug delivery, but I'm not showing that data here. So that further can enhances the therapeutic aspect of any formulation. And also the more combinations we can use in to treat not only the cancer but also other disease conditions, even in the complex diseases like diabetes and arthritis. It is already reported, but yes, we need to find out some very impactful drug combination. So I'll thank you very much for your attention, and if you have any question, just you can.
Thank you, sir. Thank you for your excellent presentation.
Thank you, ma'am.
You have explained the basics,
Yeah,
mechanism involved in cancer,
right.
how the drugs or chemicals act as an anti-cancer agent, and all those in detail. It will be really useful for our students, sir. As
Thank
such,
you.
we have few questions.
Right.
Um, the first question is: can Indian long pepper be used in bleeding disorders, sir?
Yes, actually, there are certain articles so on the Piperlongumine. So it is act as an antiplateletic, like in the heart-related problems. But for the bleeding, I don't think so. It can be used because it dilutes. It work on the platelets. So if we use the Piperlongumine for treating those kind of condition, it will not be that beneficial.
Okay, sir.
Yeah,
Our
but
next question is,
yeah,
yes, sir.
yeah. Next.
Yeah. Okay. So the next question is, can it be used in hypertension, sir?
Yes, it is already reported that it is beneficial in memory loss, Alzheimer, and even in the Parkinson. It is very useful, and even to reduce the hypertension and the insomnia. To treat the insomnia, people also use it very regularly. Yeah.
Sir, is it FDA approved, sir? Can it be used in cancer chemotherapy?
No, not till now, because the problem is for the FDA approval, we need like proper clinical trials, phase 1 and 2 clinical trial to approve the drug, any kind of drug. And these kind of phytomedicine, no one actually take much interest in that. So it's even several. I think Chinese are working on it like very vigorously. Like they are developing several formulation of it, and I hope in future they will get something. But in the not in the chemotherapies, but like combination therapies, like as a complementary medicine, they are in the use in even in the cancer treatment. They are also approved for the cancer treatment.
Yes. Is it a senolytic drug, sir?
What is the senolytic? I didn't know that.
Is it involved in senescence and all those, sir?
Yes,
Agent.
aging. Yes. Because
Yes.
as I showed you, it is targeting the P21, which is actually a cell cycle regulator. So if anything which is working on those molecules, it will help that will promote the cell senescence, like that can stop the cell cycle. So yes, that can be used for as an anti-aging agent.
Okay.
Right.
Thank you, sir. Thank you for
Thank
the
you very much.
patience in answering. Okay, it was really helpful for us, sir, actually.
Uh,
Over to Ramya again.
Thank you, sir. Thank you so much for accepting our invite and giving
thank
us knowledge on the phytotherapy, and also giving us knowledge on hallmark of cancers. Most of them won't know about the steps of
right.
those hallmarks, but it was very useful, and thank you so much, sir.
Thank you so much.
Uh, I would also like to thank the institution, Sri Balaji Dental College and Hospital, Dr. Sumati Jones, ma'am, head of the Department of Pharmacology, for conducting the series of webinar on DentoPharm 2022. Principal Dr. Jimson sir and organizing committee Dr. Satish and N.P. Murli. A special thanks to Dr. Nishad Sheikh and Angelin from DocMode organization for the technical support. And thank you for the audience who participated in the webinar today. Thank you so much.
Thank you.
Thank you,
Thank you
sir.
all. Thank you.